All right, let's dive right into this explainer. Today, we're unlocking the ultimate master recipe of molecular biology, the polymerase chain reaction, or PCR for short. Whether you're a student just stepping into the lab or a seasoned researcher brushing up on the fundamentals, understanding this process inside and out is absolutely crucial. So we're going to break down exactly how this powerful technique works from start to finish.
So the easiest way to think about PCR is that it acts exactly like a microscopic photocopier. It takes a tiny, sometimes microscopic trace of DNA and turns it into literally billions of copies. Imagine you have this massive, million-page encyclopedia, but you only need a bunch of copies of one specific paragraph on a single page. PCR allows us to target just that exact paragraph and amplify it until we have enough material to actually analyze.
Here is our agenda for today. 1. The magic of PCR. 2. Essential equipment and materials. 3. The step-by-step procedure. 4. Key considerations and troubleshooting. And 5. Expected PCR outcomes.
Section 1. The magic of PCR, the foundation of molecular biology.
Without PCR, modern applications like genetic testing, cloning, and crime scene analysis simply wouldn't exist. Often, the DNA samples we extract from a diagnostic swab or a forensic scene are just incredibly small. By using PCR to amplify those minute samples, we generate enough genetic material to confidently sequence and diagnose. It's a total game changer.
Section 2. Essential equipment and materials, the kitchen and ingredients.
Okay, so before we can start baking, we've got to set up our lab kitchen. You'll need to gather your physical tools. Primarily, that means your thermocycler, your precise micropipettes, and those tiny specialized PCR tubes. Think of the thermocycler as a highly advanced, programmable oven that can change temperatures in a matter of seconds. Your pipettes measure out microscopic volumes of liquid, and your PCR tubes are the little mixing bowls where the actual magic happens.
And here are your molecular ingredients. What's really fascinating here is how every single chemical reagent plays a highly specific, vital role. Your DNA template is the master blueprint holding the code you want to copy. Primers act as the starting blocks, telling the machinery exactly where to begin. The DNTPs, those are the raw, individual building bricks of DNA. Taq polymerase is your microscopic builder assembling those bricks. And finally, the buffer solution provides the perfect chemical environment to keep the whole shebang stable.
Section 3. The step-by-step procedure, the thermal dance.
All right, let's shrink down to the microscopic level. Sample prep requires strict sequential steps to prevent contamination before the machine even takes over. Keeping your reagents on ice is critical because it stops your microscopic builder, the Taq polymerase, from starting its work prematurely. By making a single master mix first and then allocating it, or splitting it into your individual PCR tubes, you guarantee every single sample gets the exact same ratio of ingredients. Then you just add your template and load up the thermocycler.
So, what actually happens when we crank the heat up to a blistering 95 degrees Celsius inside that machine?
Well, it kicks off phase 1, denaturation. The extreme heat literally melts the hydrogen bonds holding the DNA together, unzipping our master blueprint so we can read it. At 95 degrees Celsius, the famous double helix separates entirely into 2 single strands. If you picture DNA as a closed zipper, this heat is effectively pulling that zipper completely apart. Immediately after that, the programmable oven drops the temperature down to roughly 50 to 65 degrees Celsius. Let's call it 55 degrees for our standard recipe.
Which brings us to phase 2, annealing. This rapid cooling allows our primers to attach themselves to the exact sections of DNA we want to copy. Because the temperature dropped, hydrogen bonds can form again. The primers basically scan the single separated strands until they find their perfect complementary match, and boom, they lock on. This visual marker tells the rest of the machinery exactly where our target paragraph begins.
Finally, the temperature ramps back up to an optimal 72 degrees Celsius.
Welcome to Phase 3 Extension. This is the absolute sweet spot for our Taq polymerase. The builder grabs onto those primer starting blocks and moves right down the line, reading the single-stranded template and snapping those raw DNTP building bricks into place. By the time it finishes its run, we've gone from 1 piece of double-stranded DNA to 2.
Put it all together, and you're looking at a complete thermal cycle that takes roughly 1 minute. It happens incredibly fast. 30 seconds of intense heat to separate the strands, a quick cooling period to let the primers bind, and another brief heating phase for the polymerase to build the new sequence. Of course, we don't just do this once. The thermocycler repeats this sequence 25 to 35 times to achieve exponential growth, because the new strands created in cycle 1 become the templates for cycle 2, 2 copies become 4, 4 become 8, and so on. It ends with a single final extension step, kind of like leaving a cake in a warm oven as it cools, just to polish off any incomplete strands.
Section 4. Key considerations and troubleshooting, perfecting the mix.
Now, even master chefs make mistakes, right? So let's talk troubleshooting. If you're seeing messy results like primer-dimers, which is when your primers accidentally bind to each other instead of your target DNA, a super common optimization strategy is simply tweaking your annealing temperature. If you raise that temperature slightly, you make the environment less forgiving. This forces the primers to only lock onto their exact perfect target sequence, cleaning up your final result immensely.
Section 5. Expected PCR outcomes, the final product.
After a couple of hours in the thermocycler, it's time for the taste test. A perfectly executed PCR recipe yields billions of precise copies of your target sequence. When you take your final mixture and run it through a gel for analysis, you should see a bright, clean band. That band tells you that your primer's locked on perfectly, your temps were dialed in, and you've successfully amplified your molecular target.
Understanding PCR isn't just about memorizing temperatures. It's about mastering a foundational tool that literally unlocks the entirety of molecular biology. So, now that you hold the power of the ultimate molecular photocopier, what breakthrough will you amplify next? Thanks for joining this explainer. Keep optimizing, keep experimenting, and I'll catch you next time.